Hello and welcome all to the next lecture on bacterial staining. In today's lecture we will see PHB staining by Burdon's method, where we will use Sudan black bee stain. We will see this lecture with respect to principle, mechanism, procedure and observation. So let us begin this lecture with steps how we perform that PHB staining. For that, a smear of bacteria will be prepared on a clean grease-free slide. Then Sudan black bee stain is flooded on that slide. It will be allowed to react for 10 to 15 minutes. After reaction time, the stain is discarded. Without washing that slide with distilled water, we will pour it with xylene. Again, without washing that smear with water, we will treat it with safranin. Safranin acts as a counter stain here. Now what will be our observation? We will observe red colored bacterial cell will be observed, which is basically due to safranin. बॅक्टेरियल सेलला सॅफ्रनिन स्टेन करते PHB स्टेनिंग मध्ये and we will observe such bluish black granules which are nothing but PHB. Such bluish black PHB granules will be observed in this red colored bacterial cell. Now what does this PHB stand for? It stands for polyhydroxybutyrate. Now let us see introductory part. So these PHB granules are an example of bacterial inclusion bodies. What are inclusion bodies? They are some distinct structures which are found inside the bacterial cell. They are also called as granules. Now there are different types of granules such as polyphosphate granules, sulfur granules, glycogen granules and of course PHB granules. Now why does bacteria produce such granules? These granules act as reserve food material inside that bacterial cell. Now these PHB granules, they are lipid reserve materials. They are also called as fat granules. काय असतो या ग्रॅन्युल्सचा रोल? तर जेव्हा न्यूट्रिएंट्सची डिफिशियन्सी असेल मीडियामध्ये तेव्हा बॅक्टेरियल सेल ह्या ग्रॅन्युल्सचा वापर करतात, या इन्क्लुजन बॉडीजचा वापर करतात फूडसाठी, एनर्जीसाठी. सो दॅट ॲक्ट्स ॲज अ सोर्स ऑफ कार्बन अँड एनर्जी when bacterial cell is facing some kind of physiological stress or there is depletion of nutrients inside the media. So as stated, these polyhydroxybutyrate granules are important storage compounds and that PHB stands for poly-3-hydroxybutyrate granules. They are found inside some bacteria. Not all bacteria produce PHB granules. For example, Bacillus megaterium can produce these PHB granules. It is also seen in Azotobacter. तर PHB ग्रॅन्युल्स काय असतात? जेव्हा सेल न्यूट्रिएंट डिप्लिशन फेस करत असेल किंवा सम काइंड ऑफ फिजियोलॉजिकल स्ट्रेस एक्सपिरियन्स करत असेल त्यावेळेस हे PHB ग्रॅन्युल्स प्रोड्यूस केले जातात. सो हाऊ दिस PHB ग्रॅन्युल्स आर सिंथेसाइज्ड, हाऊ दे आर प्रोड्यूस्ड इनसाइड बॅक्टेरिया? दे आर प्रोड्यूस्ड इनसाइड बॅक्टेरिया बाय कार्बन ॲसिमिलेशन, बाय यूजिंग कार्बन सोर्सेस सच ॲज ग्लुकोज अँड स्टार्च. त्यावेळी सेल बॅक्टेरिया सेलमध्ये हे ग्रॅन्युल्स तयार करून ठेवते आणि त्याचा वापर केव्हा करते? When such easily preferred carbon sources such as glucose, they are unavailable in media. When the cell is facing a nutrient depletion, at that time bacterial cell will utilize that PHB granules. They will be used as a carbon source and energy source. Now to stain these bacterial granules, हे जे काही fat granules आहेत PHB granules, they are not easily stained. For staining them, special kind of stains are required. For example, Sudan black bee. Sudan black bee is a fat soluble stain. That's why it is used to stain PHB granules. What is principle of PHB staining by Burdon's method? Now these PHB granules, they are not easily stained by water soluble dyes. As a result, fat soluble dyes, fat soluble stains are required to stain these PHB granules. Now stains such as Sudan black bee, they are more soluble in lipid. That's why they are used for staining these PHB granules. Now when Sudan black bee is applied on a bacterial smear which is able to produce PHB granules, तर असे बॅक्टेरिया ज्यांच्यामध्ये PHB granules आहेत, PHB granules काय आहेत? They are lipid granules. आता साधा जर आपण स्टेन वापरला which is aqueous stain, then that stain is not able to get penetrated into that lipid granules. But as Sudan black bee is a fat soluble stain, it moves into that lipid material and it is retained there. As a result, these granules will be visualized after staining as a dark blue colored or black colored structures. Now let us see mechanism of Burdon's method. Now in Burdon's method, of PHB staining, Sudan black B is used. Sudan black B is a stain which is a fat soluble. As a result, that will stain lipid layer. Lipid layer is, that is, it will stain that lipid granules, PHB granules. And when we decolorize that smear with xylene, then bacterial cell will appear as colorless. So to stain that bacterial cell as a counter stain, safranin is used. Now that safranin will stain cytoplasm of bacterial cell. As a result, cell will appear red in color and these PHB granules will appear as dark blue or black colored. Now let us see requirements. For performing PHB granule staining, we will require clean grease-free slide, inoculating needle, bacterial suspension. Old culture of bacterial suspension will be required because bacterial cells which are in their logarithmic phase or which are having plenty of nutrients will not produce these PHB granules. केव्हा बॅक्टेरियल सेल प्रोड्यूस केलेले PHB ग्रॅन्युल्स? जेव्हा मीडियामध्ये न्यूट्रिएंट्सची कमतरता असेल, जेव्हा त्याला सर्वाइव्ह होण्याचा प्रश्न असेल, त्यावेळेस तो PHB ग्रॅन्युल्स तयार करेल. त्यामुळे असे सेल्स जे की फ्रेश आहेत, लॉगॅरिथमिक फेजमध्ये आहेत, ते या स्टेनिंग करिता वापरले जात नाहीत. सो फॉर परफॉर्मिंग दॅट PHB स्टेनिंग वी विल नीड ओल्ड कल्चर्स ऑफ बॅक्टेरिया. Then we will require Sudan black bee stain. So to prepare the Sudan black bee stain, we will add 0.3 gram of Sudan black bee in 100 ml of 70% ethyl alcohol. Then we will also require xylene, and last, as a counter stain, we will require 0.5% of safranin solution. Now let us see procedure. Bacterial smear is prepared on a clean grease-free slide. Then it is air dried and heat fixed. Then that smear is flooded with Sudan black bee. It will be allowed to react for 10 to 15 minutes. Excess stain is removed after 10 to 15 minutes and slide without washing it with water. It is treated with xylene. Again, it is not washed with water but it is blow dried, and at last it is flooded with 0.5% safranin. 0.5% safranin यावर पोर करायचा आणि स्लाइडला त्या स्मिअरला 5 ते 10 मिनिटाकरिता या स्टेन सोबत रिॲक्ट होऊ द्यायचं. देन जेंटली रिन्स दॅट स्मिअर विथ distilled water or tap water. Then allow it to air dry, blow dry, and by putting a drop of oil immersion on that smear, it is observed under oil immersion lens. What will we observe? काय observation आहे PHB staining by method of Burdon, that is Sudan black bee method. तर यामध्ये ब्लॅक कलर्ड किंवा डीप ब्ल्यू कलरचे ग्रॅन्युल्स मिळतील रेड कलरच्या bacterial cell मध्ये, and background will be colorless. So this was all about PHB staining. Hope you have understood the concept. In next lecture we will see flagella staining by Lefsen method. Thank you.